First of all, thanks. I would like to thank the organizing committee for inviting me, and particularly the sunset on the castle last night. It was my first time in Edinburgh. It was special. So thank you for providing cloudless skies too. The topic here, well, first the disclosures. The topic here is timely because while preparing this presentation, Francesca and I noticed that it's almost 10 days, almost to the day, since the first report of a new genetic lesion in CLL, a mutation in the NOTCH pest domain. And since then, there have been seminal papers defining the nature of the mutations. Essentially, we know now that NOTCH1 is the first, the second, among the most recurrently mutated genes in CLL. And we know now that the mutations happen in two distinct places. The first one, and the first identified, is within exon 34, where the pest domain of the molecule lies. And essentially, it's a deletion of two bases, which causes a frame shift and a stop codon. The other hotspot, mutational hotspot, is in the 3' UTR. It causes a splice variant. From the molecular point of view, the effect is similar. You have a shorter version of NOTCH1, which lacks the final amino acids. And I will tell you in a second why that is important. In the following years, many investigators are concentrating on studying which patients harbor NOTCH mutations. And again, there are several papers. I'm just quoting some of them. What we know now is that, in fact, the prevalence of the mutations increases, and that the NOTCH mutations identify patients with high risk of chemoresistance. Whether there is chemoresistance nowadays, I'm not sure, but anyway, in those years it was chemoresistance, and transformation to Richter syndrome. And the last thing we know is that if we look at time to first treatment and survival, we also notice that patients that have a clonal NOTCH1 mutations, so within above the 12% range, they have an inferior outcome, and usually they get treated earlier. So let me just recap in one slide the main features of the NOTCH1 pathway. We know that NOTCH1 is a ligand-activated transcription factor. Essentially, this means that it normally stays on the cell surface, that when it binds a ligand, there undergoes a series of proteolytic changes. Upon these changes, the NICD, so the NOTCH1 intracellular domain, goes into the nucleus, where it binds a protein which is called RBPJ, and that makes a very powerful transcriptional activator complex. So there is a gene transcription, and there's a number of genes that we associate with NOTCH1 signature. The signal is stopped by phosphorylation of the NICD and subsequent ubiquitination. And that is a very important thing. NOTCH1 signals are very timely signals. So in these 10 years, there have also been, you've seen it's almost 200 papers. There have also been many papers trying to understand what these mutations do. So trying to understand the functional implications of these mutations and the functional role of NOTCH1 in CLL. And again, I'm only mentioning a few. What we know now, to be very, very generic, is that NOTCH1 is somehow implicated in behavior of CLL cells. And what we can add, if you want to be slightly bolder, is that probably this is through fine-tuning of the B-cell receptor signaling pathway. And I will get to this towards the end. So what do we know about NOTCH1 in CLL cells? First thing we know is that NOTCH1 is always there. And this is quite comforting in a disease that has a high degree of variability. However, it's very homogeneously activated. You can see here in a work by Riccardo Dalla Favera that the NICD in cells that were taken straight out from patients is variably expressed. So even mostly in the mutated subset, but also patients that have a wild-type NOTCH have evidence of constitutive NOTCH pathway activation. However, whether it's mutated or not, this pathway is very rapidly switched off when you take cells and you culture them for 24 hours, implying that even in the presence of the mutant form, it still needs the ligand to induce its signals. And here you can reconstitute ligand activation simply by co-culturing CLL cells over cells that overexpress the ligand. In vivo, ligands should be no problem. There's abundance of the ligands within the lymph node, both of the jagged and DLL families. What is the impact of the mutation? I told you the mutation causes the formation of a slightly shorter protein that loses the PES domain, so the domain that gets phosphorylated and ubiquitinated. And this simply alters the amount of time that the NICD stays on in the nucleus. Here we generated cells with CRISPR technology, so there was no transfection or infection, that carried either a wild type or a mutant NOTCH1. And you can see that even in this very artificial model, you need the ligand. They were put on DLL expressing cells in order to see activation of the NICD. What happens is that if you then follow the cells, you remove them from the ligand and you follow them over time, what happens is the wild type cells, the signal is very rapidly shut down, whereas it stays on in the mutant cells. And gene transcription follows the same path. So essentially, it is a question of quantitative differences. The NICD stays on longer in the nucleus. And what happens? There is a competition in the nucleus for the RBPJ protein. So when the NICD is there, RBPJ turns into an activator of transcription complex, whereas when NICD is not there, RBPJ is bound by the HDAX, and that causes inhibition of transcription. So essentially, the NICD tunes this balance. If we want to see it in a different way, this is in the absence of NOTCH1. RBPJ is totally complexes to the HDAX. However, when the NICD comes in, the HDAC detaches, goes and binds the DNA methyltransferase, and this can result in methylation of promoters in some genes. You can actually visualize this by using cells that lack the NICD. These are cells that are made knockout, again using CRISPR technology, for NOTCH1. So in these cells, if you IP for DNMT3A, you find minimal amounts of the HDAX bound there, because all the HDAC is bound by RBPJ. But when you reconstitute the NICD, this time just the NICD, so that you don't need ligands, what you see is that in fact now the HDAX is gone and binding DNMT3A. And this affects promoter methylation. I can give you a specific example looking at a specific gene, which is called DUS22, and I will tell you in a moment what it does. This is the promoter methylation of the gene in conditions when there is no NICD. So all the HDAC is sequestered by RBPJ. You see that there's very little promoter methylation. However, when you put in the NICD, you can see promoter methylation. And this turns into differences in protein expression. This is DUS22, expressed highly by the knockout cells. There is no methylation in the promoter, and with reduced expression by the mutant cells, or even by the wild type cells. So in this context, I just want to mention, because it was important to us at the time, that this is part of a functional loop, because DUS22 is a negative regulator of MAP kinase signaling, and so it impacts on a number of very important biological processes, particularly on the STAT3 phosphorylation pathway, leading to a differential expression of CCR7, and thus to a differential sensitivity of these cells to migration. And in fact, if you look at the migration of cell lines, at least in mice that have mutant NICD, it's completely different to those of the wild types, and they have an ability to spread, which is increased. But let's move from a single gene to the genome. We want to know what is the genetic signature impacted by NOTCH1. So what we did is we simply compared RNA sequencing, by RNA sequencing, the expression pattern of cells that lack NOTCH1 or cells that express it in a wild type form. And you can see that there's a number of pathways that are affected. By the way, DUS22 is here somewhere, is among a set of genes that are all negative regulators of MAP kinase signaling. If you do a more physiologic experiment, and this was done again by the Richter de la Favre group, you can compare primary cells straight from patients where the NICD is expressed you have biochemical evidence of NICD activation, or you have no NICD activation. And if you compare this signature, what you see is that again you come up with BCR signaling, NFKB activation, and MAP kinases activity. So you touch on the same pathways. Now, as Francesca Ruga told you before, the BCR signaling was striking to us also because it was in line with a recent paper published in Cell Reports where activation, ligand-induced activation of NOTCH1 impacted on the transcription of a number of genes that pertain to the B-cell receptor pathway and enhanced the B-cell receptor pathway. And of course, I won't go into the data again, but what we now believe is that this may be, if you want, a truly oncogenic pathway in that this B-cell receptor may signal autonomously. It may not even need an antigen, but I don't want to get into that. And this may lead to a ligand-independent NOTCH1 activation, which again would take away the need for a ligand. I'm not sure this is true in patient cells, as we know that if we remove them from the context, so if we take them away from the patient, both these signals are shut down quite rapidly. So the question is whether there's space for dual targeting strategies. Can we take this a step forward and see it in a translational perspective? If you take cells, CLL's primary cells in vitro, and you expose them to both a BTK inhibitor and a NOTCH inhibitor, then the answer is yes, you get inhibition of both pathways, and you get significantly decreased viability. This is not only our data, but also by the Giorgio Zauli group. Is this relevant for CLL? Probably not. There is a lot of pharmacology that's still needed for drugs that target NOTCH1. They are not very easy drugs at the moment. And there's a lot on the market for CLL patients. So maybe this is not the right category. But if we select for patients who unfortunately have more aggressive diseases, then therapeutic options get less and less. And if we end up in the few patients, luckily, that get Richter's transformation, then we don't have a lot of options. So this may be a subset where to study whether we can impair NOTCH and BCR signaling together. Also because we know from published data that the prevalence of NOTCH1 mutations in these patients increases significantly. So we go from 1 in 10 at diagnosis to 30–40% of NOTCH1 mutations in patients with Richter syndrome. And for this part, we took advantage of PDXs that we just heard from Nick and Shishi. We injected, and this was done when I was at Cornell working with Rick Furman and John Allan. We injected primary Richter syndrome cells into NSG mice. About 60–70% of them engrafted, and we have now 4 different models that you see written here, where we have characterized the genetic structure. The only thing I want to point out is that when they engraft, essentially they stay stable, so they don't change over time, at least for the time points that we have followed on. One of them has the 2 nucleotide deletion in the NOTCH1 PES domain. So one of them carries the NOTCH1 mutation that we are interested in. And by the way, this is the model, it's this model here. It's the most aggressive model that we have, and it's characterized by a steady state high activation of the NOTCH1 pathway. You can see here the NICD, and it is the model that has the highest B-cell receptor activation if we look at simply biochemically. So we decided to use this model to test for the efficacy of a combination of BTK inhibitors together with NOTCH inhibitors. The way we did the experiment, as I said, it's very aggressive. Mostly mice die within 3 weeks if you inject IV, about 10 million of cells. So we did the IV injection, then we let engraftment occur over 10 days. And then we treated with a combination of ibrutinib and the APT. Again, this is not a good drug, pharmacologically speaking, and perhaps it wasn't even administered with the right schedule. In any case, we did 2 kinds of experiment. We stopped the experiment at the end, at day 21, and then we did a survival experiment. If you stop the experiment after 3 weeks, you can see that the spleens of the animals that have been treated with a combination, so blocking BTK and blocking NOTCH1, are by far the smallest. And this is because they have the lowest number of human lymphoma cells inside. This is also true if you look at histology, both in the spleen and in the bone marrow. The animals with a combination show significantly less tumor infiltration. And interestingly, if you look at survival, what you see is that, as I said, vehicle-treated animals are dead within 3 weeks. The mice that are treated with the combination had approximately a week advantage over mice treated with single agents which already have some advantage. So let me try and sum up. There have been, in these 10 years, many papers reporting many functions associated with NOTCH signaling, both in a wild type and in a mutant form. They involve chemotaxis, they involve growth, they involve metabolism, they involve resistance to apoptosis, modulation of other environmental interactions, BCR signaling. Perhaps BCR signaling is not just a slice in a pie, perhaps this is the basis of the pie. Or perhaps if you want to have a musical analogy, perhaps this is a concert for violin and a BCR is the violin. It took me like 15 years to realize that. I hope Kostas will be happy. And the rest is accompanying instruments. Last but not least, this was our Christmas party last year. The NOTCH work is mostly Francesca's work, and I'm greatly indebted to her. And these are the people with whom we've been collaborating, particularly Francesco Forconi and Graham Packen for the last B-cell receptor part. They helped us really a lot. Gianluca Gaidano and Davide Rossi got me introduced to NOTCH mutations. And I have to thank everyone in this list for providing patients, advice, and support. Thank you.
Thanks, Silvia, for showing new data in the context of the history of CLL. So we have time for questions.
I was on time.
You were. Well done, Silvia.
Very well in time, so we have time for questions, yeah. So maybe I can start off. So I do understand, of course, NOTCH2 activated in the lymph nodes and in the tissues. But I also understand if you take cells out of the blood, NOTCH1 is activated. So how is this possible? Is it because the cells are still under the influence of what they saw in the lymph nodes, or is there something in the blood?
So there are ligands in the blood. CLL cells can express NOTCH1 ligands. So in principle, there could be CLL activation. There could also be this, let's say, ligand-independent activation in those CLL cells that have a particularly activated B-cell receptor pathway. We don't know for sure. What we know is that if we take these cells out of context, so we take them, we purify them, we put them in a plate with some serum, it just goes away very rapidly. And that's independent of whether it's mutated, wild type, it just doesn't matter. And even in the cell lines, the interesting thing is that the MEK1 cell line also expresses some NOTCH1 ligands, but there's no constitutive activation. These cells, they don't show any sign of an ICD unless you plate them on top of fibroblasts overexpressing the ligand.
Okay.
I think someone...
Oh, there's one question there.
Hi, Dr. Brian Kaufman, the CLL Society USA. I'm wondering if you could comment on why you believe, or if you believe, that patients with NOTCH1 mutations don't respond as well to rituximab, ofatumumab. Do you have a sense of that, and whether that extends to all the CD20 monoclonal antibodies?
So this was a work done by Walter Gattei here in the audience. And again, it's linked to the fact that there are different levels of CD20 expression in patients that carry NOTCH1 mutation. And again, it's a genetic regulation on the CD20 promoter. And so it's linked to the fact that if you have lower levels, you don't reach the threshold to see cytotoxicity. That was noted in a trial where, with a comparison of FC versus FCR.
Good, okay, thanks Silvia. Thanks again.